Everyone, welcome to our last seminar of the year 2022. We'll pick up again sometime in the spring. I don't remember exactly when our first one is, but we'll be sure to send it around well in advance so that everyone can show up. Today, it is my pleasure to introduce Calvin Yee, who comes here from the University of Georgia, where he's an assistant professor in the Department of Genetics and also bioinformatics, correct? Yes. Mm-hmm. So Calvin did his undergrad work in Wuhan, China, and then he went on and did his PhD in nutritional genomics at Cornell University. He stuck around Cornell for a postdoc where he shifted from nutritional genomics to developing a lot of computational and statistical skills in the biostatistics and computational biology department. And I think it's pretty fitting that he's been working on he's sort of in his research now, he's combined a lot of his training that he gained at Cornell specifically, right? Looking at combining nutrition and really cutting edge computational and statistical tools to investigate gene and environment interactions, which is the focus of his research. And for the most part, I would venture that the environment that Calvin's been studying the most, or the variation in the environment that he's been studying the most, is variation in what people are eating, their diet and their nutrition, and how that impacts a range of functions in people, in particular metabolic diseases. And with that, I'm going to hand it over to Calvin, who's going to tell us about some of his work. Thanks so much.
Thank you. Thank you very much for the introduction and for inviting me to visit this center. I wanted to visit this center for quite some time because of my interest in evolutionary medicine. So my interest in evolutionary medicine dating back to my undergraduate years when I read the book, Why We Get Sick. And I was always interested in evolution, and that book convinced me that we can use evolution to think about how we maintain our health and treat disease. And that idea stuck with me, and it influenced my research interest, and now it influenced how I set up my research program at UGA. As Noah mentioned, right now I center my lab on nutritional genomics or nutritional genetics. I thought about using evolutionary medicine for my lab logo, but I feel it was too broad. For nutritional genetics, we are mainly interested in two research questions. First is how evolution shaped our unique nutritional requirement as a human individuals compared to other species. And also when you consider the genetic difference across each one of us, how does our genetic background make us unique, and how can we get those information to use when we think about what we should eat for our nutrition. I decided to have one slide talk about the major research going on in my lab. So the focus is nutritional genetics, nutritional genomics. We focus on one group of nutrient, the omega-3 and omega-6 polyunsaturated fatty acid. It's basically when you take fish oil, you go for the omega-3 fatty acid. We have different research direction. The first one is evolutionary genetics, and that was my training background in population genetics. I studied how different human population adapt to their local diet, and now I expand a little bit to compare human to other species to think about the unique human nutritional requirement. My background was mainly computational, but when I studied my lab at UGA, I decided to start a wet lab. So we're doing experiments in human cell lines so we can really figure out the causal variant and molecular mechanisms. In the last few years, I spent a lot of effort to develop a direction called genetic epidemiology, or just quantitative genetics. For all those nutrient level in the blood, it is a trait that is controlled by our diet. It's also controlled by our genetic background. It has a genetic basis. So we perform genome-wide association study to find the genetic variants associated with, for example, the level of omega-3 in our blood. We mainly work on gene-diet interaction, and I'm going to share some study on that. We also spend quite some time working on Mendelian randomization analysis. Mendelian randomization is a statistical technique that supposedly can help us infer causal relationship between an exposure and a disease. And for nutrition, we have so many different kinds of nutrients. And there are so many claims out there that this nutrient is good for that. But we don't really have large-scale randomized control trial to test that. So my hope is that to use some genetic technique to use the shared genetic basis between different traits to help us understand the role of those nutrients in different diseases. And also in general, we're doing some omics data analysis, some single-cell RNA-seq analysis in the lab. And it has been a lot of fun running my own research program. And one thing being as a PI, one very nice thing is that before, when you are a grad student and a postdoc, you can work really hard every day, but you can only manage 1 or 2 projects. Now as a PI, you can recruit students to different projects and can see different projects moving in parallel. It's very satisfying. So for today, I'm going to share with you 2 major projects. The first one is an evolutionary project about the FAST gene. It was mainly done in my postdoc, but that's where I studied all my research program right now. The second major direction, I have listed a few projects here, but I'm mainly going to focus on the first one. It's a GeneEnvironment Interaction project. So I'm going to start with the first project, the adaptive evolution of the FAST gene. What are the FAST genes? The FAST genes actually is composed of 3 genes, FAST1, FAST2, and FAST3. They locate right next to each other on chromosome 11. And FAST1 and FAST2 has very well-known function. They are involved in the biosynthesis of the long-chain omega-3 and omega-6 fatty acid. So omega-3 and omega-6 fatty acid are 2 groups of fatty acids that share similar chemical structures. They have relatively long carbon chain, ranging from 18 carbon to 24 carbon. And they are also called polyunsaturated fatty acid, means that they have 2 or more double bonds. And based on where the first double bond is, if it's like the 6th carbon from the omega N is omega-6 fatty acid. If it starts at the 3rd carbon, that is omega-3 fatty acid. So you can see that it's actually different individual fatty acid. Sometimes for those that has 18 carbon or shorter, we call them short chain or medium chain. For those that are longer, we call them long chain. But it's a lot of confusion there. It's not fixed. The first 2 fatty acids, LA and AOA, are considered as essential fatty acids because your body could not synthesize those 2 fatty acids. You need to eat them from your diet. For all others, you can synthesize them using the first 2 essential fatty acids. And FAST1 and FAST2 are the rate-limiting enzymes in this pathway, and that's why they are so important. The physiologically active or more important ones are the long-chain fatty acid, AA, EPA, and DHA. DHA and the AA right now are commonly supplemented in infant formula because they play very important roles in brain development. When you take fish oil supplement, you mainly go for the long-chain omega-3 fatty acid. In terms of dietary source, you can get, when you eat a lot of red meat, you mainly get the ARA, AA, omega-6 long-chain fatty acid. When you eat a lot of seafood or fish, you have the EPA and DHA. For the 2 essential fatty acids, they are also called the plant-based fatty acid because we mainly get them from a plant-based diet or all those plant oils. So now when you think about people living on different kind of diet, for people that live on a vegetarian diet, you do not have access to those meat product or seafood, then you only really have the essential short-chain fatty acid. You need to rely on your body to give you the long-chain important ones. So you can imagine that genetic adaptation in those populations that rely on plant-based diet, those mutations that enhance this biosynthesis process is adapted. On the other hand, for people that rely more on seafood, like the Inuit, they do not really have plant. Then they have a huge amount of long-chain fatty acid already from their diet. So you can imagine that they do not need any more variation to help them enhance the biosynthesis process. So based on the understanding of this biosynthesis pathway, there are 2 evolutionary hypotheses. In those populations that rely on plant-based diet, mutation that enhance the biosynthesis is adapted. In contrast, for those populations that rely more on seafood, then mutations that reduce the biosynthesis may be adapted. And those are the evolutionary hypotheses I tested in my study. But this is not how I get involved in the FAST gene. Like this is clean and nice. We stay in a pathway, you come up with a hypothesis, you test it, you get something. It wasn't how this story happened. The reason I got into the FAST gene was because of my committee member, Dr. Tom Brenner. Tom has been working on the FAST gene and the PUFA for his whole career, 30 years at Cornell. And he was in my research committee. He was very influential for my research. But I didn't really work with Tom until the last month of my PhD. I was hanging around at Cornell. And Tom has been working on the FAST gene, and he has a special interest on a variant. It's an insertion deletion polymorphism in the FAST2 genes. He, based on cell line experiment, has already shown that the insertion allele is associated with high expression of FAST1. He also collected some sample by himself to show that the insertion allele has higher frequency in the Indian compared to some US samples. And we know that the Indians traditionally rely more heavily on plant-based diet. So Tom came up with this hypothesis that the insertion allele may have adaptive advantage in India. He sent this data for peer review, and one of the reviewers was asking, what are the US samples? You collect the sample from the US, but US is ethnically diverse. What are they? And also, can you check the frequency of this variant in the 1,000 Genomes Project? For those that don't know about the 1,000 Genomes Project, is a international collaboration that collected over 2,500 genomes from different from 26 populations. So it was a resource. You can go there and check the frequency of a specific variant. So Tom asked me to do it. It was the last month of my PhD. It seems simple. I just did it. So I get the figure and gave it to Tom. And this is what I got in the 1,000 Genomes Project. And it broadly is consistent with Tom's data. The insertion allele here, the insertion allele in red in this figure is consistent, has a very high frequency in the South Asian, and the insertion allele has much lower frequency in the Europeans. So most of the sample from the US is the European sample. I gave this figure to Tom, and I was staring at this figure, and I told myself I can do better than just giving him this figure, because I was training population genetics. And when you have a variant that has very different frequency across population, you can guess that there's selection. But there's formal tests you can do, right? You can do a bunch of statistical tests to see if there's selection in South Asian or in Europeans. And that's what I did. It was a side project for my postdoc. It took off, and it took all my time for my postdoc. So I used the data from the 1,000 Genomes Project. I performed a bunch of population genetics tests for the presence of positive selection, and I list different class of the selection tests here based on side frequency spectrum, haplotype homozygosity, population differentiation. So I do not want to go into detail here. I just want to share with you one key result. So when I look at the South Asian, I performed the haplotype homozygosity-based test. And this is one of the key figures for the insertion deletion polymorphism. And so this is called haplotype bifurcation plot. It may be for the older generation, but the way you read this plot is that each line represents a haplotype. And we separate the haplotype based on which allele they carry at that specific variant of interest. We separate to haplotype carry the ancestral allele, haplotype carry the derived allele. The thicker line represent the higher frequency of the haplotype in the population, in that specific group. And this is the signal you're looking for, a haplotype that has very high frequency in the group that carry a specific allele, suggesting that this derived allele is under selection. And this is another plot to test from this point of interest, going to two directions, for how long the homozygosity maintain. If an allele is under selection, it's able to maintain the homozygosity for a longer distance compared to the other alleles. and can quantify them using the IHS and the NSL statistics, and both of them are highly significant for this variant, suggesting that this variant and the variants nearby has adapted signals in the South Asian, and that was, and we have many other tests to they are consistently showing that surrounding that region, the FAST gene has adapted signals in the South Asians. So that side project turned into a very nice publication. And for this first project, we mainly used the 1,000 Genomes Project data. At that time, it was around 2016. It was at the high time of ancient DNA, and we're thinking, can we use ancient DNA to study the adaptive signal of these specific alleles? And majority of the ancient DNA were mainly located in Europe or Western Asia. So we look at those regions. We collected ancient DNA for 3 groups: the pre-Neolithic hunter-gatherer from 4 different time points, and then the early European farmers and the pastoralists. And those 3 groups represent the 3 major ancestral groups for the present-day Europeans. So using those ancient DNA, we can test the presence of adaptive evolution. And the idea is relatively simple, because once you have allele frequency across different time points, you can just simply check if there's an increase in frequency for specific alleles. So I apply an existing method that takes into account admixture across 3 populations and then compare to the frequency in the present-day populations. And I was eventually it turned into a statistical p-value for each variant. Is there a statistical evidence that a specific variant has increased frequency during that time span? And that time span is about 8,000 years to present date. And the p-value here I show the negative log 10 p-value. The higher the value here, suggesting more statistical support for the presence of selection. And so this is very similar to a Manhattan plot in GWAS. You can see that there is a cluster of signals centering around FAST1 and FAST2, suggesting that there's selection signal in Europeans. It's mainly in Europeans. The previous study we looked at South Asian. Now, because we use ancient DNA, it was mainly in Europeans. And here I just highlight one of the top SNP located in the FAST2 gene. I also highlighted the indel. And the indel do not have signal at all in the Europeans. And there's some complication with the indel because it's very difficult to do genotyping accurately for the indels. But overall, there's signal of adaptation in the Europeans. And when I look across different populations in the 1,000 Genomes Project, just pick one SNP as an example, but the pattern is the same for many SNP surrounding that region. If you look at the haplotype, we can see the same frequency pattern. The allele A has very high frequency, has the highest frequency in the South Asian. It has about 60% frequency in the Europeans. But that same allele is not present in the Inuit. And in a previous study, they have already shown that the FAST gene has adaptive signal in the Inuit, likely adapted to their seafood-based diet. So based on my study, the 2 population that are farming population rely more on plant-based diet. They both have adaptive signals for the FAST gene. And then in the Inuit, they have opposite adaptive signals. And sometimes to make it easier, I call the allele A in the farming population as farming adaptive allele, the other allele as hunting or seafood adaptive alleles, just to make it easier to remember. Otherwise, A, C, or T is a little bit confusing. And then using the ancient DNA, because I have a time series data for the ancient DNA from dating back to 30,000 years ago to present day, I can see how does the frequency change over time for that haplotype and then I just pick one SNP to represent this trend. And you can see that for the farming adaptive hap alleles, we know that in the recent time, in the last 8,000 years, it was increasing frequency. That was consistent with my test using modern DNA alone. If you look back in time, like from 30,000 years ago, the frequency was going down to close to 0 about 8,000 years ago. On the other hand, the other allele is increasing in frequency because they because there's just two alleles. And so you can see that around this time, there's a change in the frequency trend, and that time corresponds to the agricultural revolution. And then with the agricultural revolution, the population also switched their diet. Before the agricultural revolution, the population relied more on hunting, animal gathering, and they eat more seafood, more diverse diet. Compared to after the farming population really survived on a few staple food and more plant-based diet. So those are the evolutionary analysis showing that the FAST gene has adaptive signals in different populations, farming population, seafood-based population, and then also across different historical time periods before and after the agricultural revolution. So those were all evolutionary analysis. And then with the evolutionary signal, one natural question is that what was the molecular mechanism? Does that fit with our understanding of the physiology of the fatty acids? So we look at the molecular mechanism for the adaptive alleles. And we use the EQT analysis, basically testing the association of genotype with gene expression. We show that the farming adaptive alleles is associated with high expression of FAST1 in liver and in most tissues. So the adaptive alleles will increase the conversion of the short-chain product to the long-chain product. And then we also look at the association of the genotype with all these fatty acid levels in the blood. And we found that the farming adaptive allele is associated with higher level of the long-chain product and then lower level of the precursors. So it's consistent with the higher expression of FAST1. So with the adaptive allele, the farming adaptive allele, people eating plant-based diet, the FAST1 expression was increased. So increased the conversion of those plant-based fatty acids to the long-chain fatty acids. The same gene is also associated with many traits, including IBD, inflammatory bowel disease, some cancers, some bipolar disorders, and I also recently checked the association with many phenotypes using UK Biobank, and I summarized them here. One of the SNP is associated with many traits, and majority of traits is related to blood lipid and DHA I just showed. So the FAST gene is hugely important, connected to many biomarkers and many traits. The FAST gene was also adapted across human history to a specific, potentially to a specific kind of diet, suggesting that in present days, for people that have different genotype at that position, if we have a different kind of diet, it may impact our health differently. In summary, so here are my overall thought. It's also my schematic when I think about my research program, is that we start with an evolutionary project or evolutionary analysis. We look at people that live on different environment, in this case, different diet. Over time, there will be genetic adaptation adapt to a specific diet, and that genetic mutation become more common in a specific population. If you have a different diet, you have a different genetic variation that is adapt to that diet. And this is kind of like a gene-diet matching evolutionary process. The implication is that nowadays, we should try to match our diet to our gene, just like nature did over time. So this is a very appealing idea to me, that we can learn from evolution about what kind of diet to eat. But sometimes I'm staring at this and I ask myself, can I find something to support this idea? So yeah, I have the evolution stuff, but do I have more? Like when you try to recommend someone, like you should take this diet, do you have more evidence? So I'm telling myself the best experiment I can do is that I recruit participants, genotype them, and then based on the genotype, I assign those that I put them on different diet, and then I follow them for a long time, and I see how do they perform. But you know that randomized control trial, put someone on a diet for a long time is not going to happen. And then And then I was thinking we can use some existing cohort data. If we can find a sample that live on a specific kind of diet, for example, find vegetarians, and then we find those non-vegetarians. They follow two different kind of diet, and there's natural variation in those samples. Can we see a difference of the effect of the genetic variant? For example, for the fat gene, the same variant, do they have different disease association in the vegetarian and in the non-vegetarians? I can follow them. I can do another phenome-wide association analysis and hopefully try to find different associations. In vegetarian, you have this association, but you do not find them in the non-vegetarian. So this figure is only a schematic. It's an idea. I haven't generated this figure yet, but this gives me the idea of testing context-dependent genetic effect, or more formally, it's also called gene-environment interaction in genetic epidemiology. And that's what I started to work on in the last few years. So that's one of the major directions. I'm also doing some comparative genomics to study the evolution of fat gene across different species. I haven't had mature result yet. And so for this project, we mainly focus on the one gene, but are there other genes related to omega-3?
Choose the right audio apps for you. Which one do you want to use to listen to this?
Oh, sorry. Sorry, Siri. Yeah. And also, can we study the omega-3 as a complex trait? How does that complex trait evolve over time? And for that to happen, we first need to find all the genetic variants associated with omega-3. And that's what we are working on. And the last one is just molecular experiments. So that's the first part of my research. For the next part, I'll focus on gene-environment interaction. And I want to start from the very basic to make sure that you understand the concept of gene-environment interaction. So in a typical GWAS, you find participants that have different genotype, 3 possible genotypes. We code it as 1, 2, 0, 1, and 2, and see if they have different specific phenotype like height, like weight. So that's a typical genome-wide association analysis. You found a genetic variant associated with the trait. In a typical epidemiology association analysis, you're interested in a specific exposure, like for example, taking fish oil supplement. Is it associated with lower triglyceride? So that you just do a simple association analysis between those two groups. When we do gene-environment interaction, we are basically doing stratified analysis. We stratify the participant. For example, we stratify them by their environmental exposure. In this case, we separate into having no exposure with the exposure, and you can see that the genetic effect is parallel to each other. You can do the same thing, stratify them by the genotype, and the environmental effect is consistent in different genotypes. In this case, there's no interaction. The effect of the gene and the effect of the environment, they are linear. They're just additive to each other. Another way to think about it is that we can think about the genotype as a reference level 0. If you increase your genotype by 1, you increase your phenotype by this specific amount. And then the effect of the environment is this amount. If you have the gene and the environment, then you increase, just add them together. So in this case, there's no interaction. And then I'm going to give you one example that has interaction. In this association analysis, the genotype appear to have no association with the outcome. There's some association for the specific environmental exposure. And then when you perform the stratified analysis, you can see that in the no exposure group, there's a negative relationship, and then in the exposure group, there's a positive relationship. So those two groups cancel out each other when you combine them. Same thing, you can stratify by genotype, and you see that the effect of the environmental exposure is different across different genotype groups. So in this case, the effect of the genotype depends on whether or not you have a specific environmental exposure. So in this case, when you have a specific genotype and the environment, the effect is no longer linear combination. You need an additional term to account for that interaction. So in we can perform some simple statistical analysis to find those interaction. And so this is a typical model in a typical regression model. To identify interaction, you just add an interaction term in that regression model, and then you can look at if this specific parameter is significant or not. Sometimes people also do a 2-degree freedom test to jointly consider this term with the genetic term. And the idea is that if this effect is weak, but you combine those two of them, you can still find them. So that's the method I use when we study gene-environment interaction. There's more advanced method, but this is the basic one. The promise of performing this kind of interaction is that you can quantify the nonlinear effect, putting gene and E together. You can find some new variant for the trait, because usually when you mix them, you can even out the effect. And then we can define specific responder or non-responder to a specific environmental exposures. The challenge for gene-environment interaction is the first challenge is the sample size. The current estimate is that you need 4 times the sample size as a typical GWAS, and for GWAS, we are already using tens of thousands of samples. And also for those big samples, you need to have high quality genotype, phenotype, and also environmental information. Majority of the cohort do not collect high quality dietary information. Luckily, things are getting better because now we have some biobank scale data like the UK Biobank. To those that are not familiar with UK Biobank, it's a large prospective cohort that was recruited more than 500,000 participants back in 2006 and 2010, and they have been following the cohort until nowadays. They have genotype data. They are doing whole genome sequencing right now. They have electronic health record. They measure a bunch of biomarker and metabolomics data. And also very nicely, it's very open to the scientific community. There's an application process, but it's relatively smooth, and it's very convenient to get access to the data set. And I should really give a shout out, thank you to that participant and the managers of the data set. My lab right now really survived on this data set. And you can see how many things we have done using this data set. The dietary information, there are 2 dietary questionnaires in the UK Biobank. The first one was collected at baseline. Almost everyone have that questionnaire. It was not that detailed, so in some sense, it's not that useful. The second questionnaire is a 24-hour dietary record. It collects more information, but it was only administered to 200,000 participants. In our first study, we look at fish oil supplementation because of our interest in omega-3 fatty acid. And also fish oil is already known to be correlated with blood lipid levels. And blood lipid level as a continuous trait can also increase statistical power in an interaction analysis. We just put the 2 questionnaires together, and we only include individuals that answer yes, taking fish oil to both questionnaires, answer no, both to both questionnaires. So that hopefully give us a higher quality fish oil supplementation information. And then we perform the analysis in UK Biobank. We use UK Biobank as a discovery, and then we use another smaller cohort, very small, only 7,000 as our replication data set. And then we put the 2 cohorts together to perform a meta-analysis. At the end, we found 2 loci that has interaction for LDL, 1 loci for HDL, and then 1 loci for triglyceride. And here I just give you more information about the triglyceride loci. So this is a local Manhattan plot for that triglyceride loci. A cluster variant here has statistically significant interaction with fish oil to impact triglyceride. So it may be easier to visualize the effect in this bar plot. We stratify the participant based on their fish oil status, and then you can see that in those that are not taking fish oil, the G allele is associated with higher triglyceride. In those that are taking fish oil, G allele is associated with lower triglyceride. The last group has too much standard errors. Another way to visualize this result is stratify them based on their genotype. For the AA group, there appears to be no difference between those that are taking and not taking fish oil. In the AG group, taking fish oil is associated with an apparent decrease in the triglyceride. So that's what we mean by interaction. Like the effect of gene depends on the diet. The effect of diet depends on the genotype. So yeah, we found that the fish oil modified the genetic effect, and with or without fish oil supplementation is different. So this just repeat what I just said. So we found this variant, and similarly, we always ask what is the candidate gene in that region. And in this case, we look at same thing, look at the regulatory signal, EQTL signal in that region. And the same SNP here are also associated with the expression level of GJB2. It's only with this gene. It's not associated with the other few genes in this region, suggesting that GJB2 is likely one of the candidate gene in this region. And this is for one of the top SNP. You can see that the AG allele is associated with higher level of GJB2. And GJB2 is a encode for a gap junction protein, and this specific group of protein previously in human cell line and animal model have been shown to be respond to omega-3 supplementation. So it seems plausible that GJB2 is one of the candidate genes in this region, but we do not really look into the molecular mechanism. In summary, for this study, we found a few genetic variants that modify the effect of fish oil on blood lipid. We still don't know the molecular mechanism or mechanistic explanation. We still need a replication in a bigger data set. To me, I feel that it's a very small number of interaction. Like, is it possible that it was because we have a small sample size? If we have more sample size, can we find more? And can we also try different environmental factors to find more interaction effects? And the next project is related to the project I just presented. So the project I just presented is looking at each variant one by one across the genome. For each variant, I apply this interaction model and ask if that variant modifies the effect of the fish oil. It's possible that for each single variant, the effect is very small. Can we somehow put all the variants together and do a single test? And that's what we are trying to do, because for blood lipid, there has been several large GWAS that are performed for those blood lipid, and many genetic variants have been found to be associated with blood lipid. So supposedly we can develop a polygenic risk score or polygenic score to predict our level of different blood LDL, HDLs. And in most of the GWAS, they did not consider environment-specific effect. So we can use those GWAS summary statistic and then develop a polygenic score of, for example, triglyceride. And then we can put the polygenic score in the interaction model to see if the polygenic score of blood lipid interact with fish oil interaction. The idea is that if fish oil is able to modify your genetic potential for blood lipid, then the GWAS summary statistic we have right now, the polygenic score will predict observed lipid differently in people that are taking fish oil and those that are not taking fish oil. And this is the result for the total cholesterol. Each dot represents one individual. The x-axis is the genetic score for basically the predicted total cholesterol level. We normalize them. And then the y-axis is the observed total cholesterol, normalized to the unit of standard deviation. And we know that the genetic score is able to predict our observed lipid level. Not very well, but the R-square is about 0.3, but we are able to predict that. And then we stratify them into two groups. They are taking fish oil and not taking fish oil. We perform a linear regression, and you may laugh if I try to convince you that those two lines are different. They have different slope. We quantify them in the two group, fish oil intake and no fish oil intake. In the fish oil intake group, every unit increase in the genetic score is associated with 0.3 unit increase in the total cholesterol. In the group that are not taking fish oil, the magnitude is slightly higher. We did the same thing for the other three lipid, and you can see that statistically it's significant. There's different effect for the polygenic score across the two dietary supplement group. And another nice thing that convinced me that this result may be real is that for those that are taking fish oil, like they have less increase in the bad cholesterol. You can say the total cholesterol, LDL, triglyceride. Like if you have a high score, you have a high increase, but the magnitude increase is less than those that are not taking fish oil. On the other hand, in the HDL, considered the good cholesterol, it increased more. So it seems that taking fish oil can shift your lipid profile slightly towards a better lipid profiles. So that's another study we did to show that the fish oil supplementation may modify our genotype for blood lipid. And at the end, I just quickly run through, do similar analysis for vegetarianism. We are interested in PUFA, so we did the largest PUFA right now in the UK Biobank. We find many genetic variants associated with PUFA for sure. And we find many novel loci. And then recently we performed an interaction analysis using vegetarianism as the environmental factor. And we were able to find 3 significant interactions. Right? And this is just an example, the top SNP here in the non-vegetarian group. The SNP is not associated with LA, the one called the omega-6 fatty acid. But in the vegetarian group, there's a linear trend that the genotype is associated with LA. So that's another example interaction. And we also test gene-vegetarian interaction on 30 biomarker for different disease. And we found another 3 significant interactions. And this is just one called the example that for calcium. For this top SNP, in the 3 genotype, the effect of vegetarianism is different. In the CC genotype, being a vegetarian is associated with lower calcium. But in this genotype, being a vegetarian is actually associated with higher calcium. So that's another example of interaction. Overall, based on our GeneEnvironment Interaction study so far, we found some example, but honestly, not as many as I want. It may have to do with the sample size. For the vegetarian, after QC, we only really have 2,000 vegetarians. But we're having more. We are finding more and more examples that, based on cohort study, we can find vegetarian or gene-environment interaction. And nowadays, I'm just thinking about, so we have 2 approaches to study gene-environment interaction. Can we use this kind of approach in the cohort to identify variants that was historically adapted in human evolution? Like, for example, gene-vegetarian interaction, we found some genetic variants that has a specific effect in the vegetarian group. Are those adapted in the farming population? Eventually, I'm hoping to put those 2 approaches together to give me some more confidence in recommending people to take a specific diet that are matched to their genome. Okay, so that hopefully I will have more concrete stories in the future. And with that, I want to thank the grad students that performed most of the study, and the gene-vegetarian interaction in the PUFA was done by 2 undergrad in the lab. And this is my research group right now at UGA, and thank you. I'm happy to take any questions.
Do we have some questions in there? Yeah, Ben, take it over. Pass the mic around.
Great talk. Thanks so much.
Thank you.
So I was just wondering, with the UK Biobank, you also have access to other data like physical activity.
Yes.
Could you control for physical activity in like the omega-3, the fish oil supplements?
Mm-hmm.
I'm just thinking that people who take fish oil might be healthier in general and might be more like active and more engaged in physical activity. And could that also be impacting the results as well?
Yeah, that's a great question. We did not control for physical activity in this specific study, but I agree that it could totally be a confounder in that analysis. One challenge in all this gene-environment interaction analysis is that we include one environmental factors, but as you mentioned, it is correlated. A lot of very healthy lifestyles are correlated. So for the variant we found, actually, I do not have confidence that the fish oil is the causal environmental factors. And actually, in all the association analysis, I do not have confidence about causality at all. We have the physical activity data, and in one of our recent study, it wasn't a gene environment interaction. It was more a typical epidemiology association analysis, testing the association of omega-3 level with mortality. In that study, we included physical activity. We include a lot more lifestyle factors. And we were able to show that for people that have high omega-3 level at baseline, followed in the last 12 years, people have high omega-3 tend to have less mortality. So omega-3 in that study seems to reduce mortality risk. And I agree with you, it's very difficult to tease apart which one is the causal lifestyle factors.
What about BMI?
Oh yeah, BMI. In all the analysis we did, we test BMI separately. We do not include BMI in our original model because nowadays BMI could be a confounder, but it could also be a collider. Is that BMI is the downstream effect of fish oil supplementation and also another disease that we're testing. Then BMI is a collider. Instead of BMI impacting whether or not you take fish oil, BMI impact your disease risk. In that case, it's a confounder. For confounder, you need to control for it. For collider, if you control for it, you may induce a selection bias, or sometimes called collider bias. They will induce false positive. So what we do is that we usually test two models, with or without BMI, and then see if there's any big difference. In majority of analysis we did, the BMI do not make a difference. Okay.
Great talk as well. You may have mentioned it, and I may have missed it. When you're doing the complex models, when you're calculating the polygenic risk score, what were you how do you get the polygenic risk score? What are you calculating the gene against in that instance when you're looking at when you're trying to bring in what was the outcome variable, and how are you figuring out what things to put in the polygenic risk score?
Cool. Yeah. Thank you. Yeah, I didn't go into the detail about the polygenic score project. So for polygenic score, for example, for total cholesterol, there was an existing GWAS that was done for total cholesterol. So it was a very big GWAS published in 2021. And like most GWAS, they included population structure, covariate. And then for each SNP, they have a effect size for each SNP. Basically for each SNP, for every effect allele, how much they increase the total cholesterol. And there are different algorithms to calculate polygenic score. The essential idea is that you look across the genome, you pick SNP that are independent of each other. Each SNP has an effect size. Right? And then you go to a new individuals, you look at their genome, like this SNP, how many effect allele they carry. If they carry 1, you add by beta. If they carry 2, there's 2 beta. Right? And go across the genome, add them 1 by 1, and get one number. And that number is the polygenic score we used. Each individual has one polygenic score. And then we put that polygenic score into a linear regression model, including the interaction term. We included a bunch of covariate, BMI, Townsend index, genetic ancestry, and statin. Yeah, we include that. Statin attenuate the signal a lot, but it's still significant. The result I show you has statin in the model. Yeah, so that's the model we use. We use the previous GWAS, calculate the score in UK Biobank. The previous GWAS do not include UK Biobank. We explicitly use the GWAS that do not have UK Biobank so they doesn't bias our result. Get those summary statistics, go to UK Biobank, calculate a score in UK Biobank. And UK Biobank also measure the blood lipid. And then we're testing the association between the score and the measured blood lipid. We stratify them to see if those associations have different slope. Yeah.
I'm gonna ask a question, then I'll hand it over. Marina had her hand up first, then you guys. It's related to a question that was posted on the chat, which I also wrote down, which is good. Which is when talking about the FADS genes, your first hypothesis. It makes sense that there might be, I think you know where I'm going. It makes sense there might be adaptation for this allele to increase biosynthesis in those who are eating, vegetarian diets. But why would there be selection against that allele in individuals who, why would they need to downregulate biosynthesis, even if they are eating meats?
That is a really good question. And I was scratching my head. I was thinking really hard about that question. Is it possible to have too much omega-3? Is it possible that having too many omega-3, omega-6 is bad? And I read through a bunch of literature to see if there's an upper limit for omega-3. Really not much. Having too much omega-3 will increase your bleeding risk. That's one of the side effects. But there's really no convincing evidence for me that having too many omega-3 is bad. So I'm just waving here that you're wasting your energy synthesizing the omega-3 that are not necessarily there. Yeah, and there's a lot of follow-up study on the FADS gene locus from the evolutionary perspective using ancient DNA. And the selection in the Inuit may not be unique to the Inuit. If you look at the hunter-gatherer population across the globe, the Indonesian pygmies and the hunter-gatherer in the Southeast Asia, they actually have the same haplotype as the Inuit. So actually, the selection may happen way before the Inuit reach that area. And in Africa, when you do look at ancient DNA, Africa right now has the farming adaptive allele. So in that sense, it's similar to the European and the South Asian. But I show you that when the selection happened in European, it was the hunting adaptive allele. So when they go out of Africa at that time point, it must have some I think it must have some hunting adaptive alleles. So they right now the idea about FADS gene is that there are three phases of adaptation. At first, the farming adaptive allele, the vegetarian allele, goes up in African, and then at some time point, it goes down in the process of going out of Africa. And then in the farming population, it goes up again. And about why, we don't know. Why in the hunter-gatherer, the vegetarian allele goes down. And I actually do not have a good explanation for that. That's the fun thing about evolution, right? You just keep thinking, what would be a good evolutionary explanation? Yeah. You have to do that in evolutionary study. Is the fun of doing evolution is that you can come up with creative explanation. Like sometimes also a little bit dissatisfying is that I could not show that. Okay.
Yeah, great talk, and thank you. This answered one of my questions. And my other question is, I know that obviously G/E models need a lot of power typically, and that's very challenging to get. But one way to potentially increase that is, of course, with finer grained data. So I'm curious if you're interested and have started to try to look at in any way the duration that people have been on a vegetarian diet or taking supplements by having people who are on kind of longer duration or have environmental exposures for longer amounts of time, we might see larger effects. So
Yeah, it's a great idea. So instead of increasing sample size. We may think about increasing the duration of a specific dietary habit. It's a good idea. In the analysis we did, we took advantage of the 2 dietary questionnaire to make sure that it is consistent in 2 dietary questionnaire. It helped a little bit. And actually, we in the vegetarian analysis, we try 2 analysis, sensitivity analysis. We try using 1 questionnaire, the self-reported vegetarian, and then we try using 2 questionnaires to combine them. And by combining 2 questionnaires, we have a smaller vegetarian group. And we found more signal with that smaller group. Supposedly, it was because of a higher quality vegetarian. But I have no proof to that. It's small sample size, we still find more significance. So maybe higher quality. But the limitation is that I do not have access to those large cohort data. So if you are aware of any data that are easy to get access to, let me know.
Two more questions. Okay.
Hello? Okay. You said a lot today, so.
Yeah, sorry.
So FAD2 had the insertion, right?
So the insertion, is in one of the intron region of FAD2.
FAD2, when it has higher expression, FAD1 has lower expression?
I didn't talk about FAD2. I only talk about FAD1.
Oh, FAD1, my bad. Okay.
Yeah, but you may lead to a good question, but let's focus on FAD1. When I show all the association with gene expression, I only talk about FAD1. So the insertion allele is associated with higher FAD1 expression, and the insertion allele is associated with the farming adaptive allele, right? So farming adaptive allele increase FAD1 in most tissue.
Increased expression, FAD2 decreased?
I didn't talk about FAD2.
Yeah. Dude, is there?
Oh, so that's the question.
Yeah. Okay.
Why do you guess FAD2 decrease?
I don't know. I was just wondering because if something increases, maybe something else.
Why? They were in the same pathway.
Oh.
They were in the same pathway. Why FAD2 decrease?
Okay. Never mind.
No. It's a good question.
I don't know. It's just something I was thinking.
So I want to go back to that slide because it's actually a really nice question, and I didn't show it. I didn't show the result for FAD2 for a reason, and since you mentioned it, why not just talk about it? Yeah.
I feel lost now.
No. So you're talking about FAD2, right? And here I only show result for FAD1. I did not talk about FAD2 at all.
My bad.
I neglected. But when you look at this figure, why would you guess FAD2 decrease?
Well, I was thinking people with certain, I guess.
If you have the farming adaptive allele, you want to increase your biosynthesis. So you increase FAD1, makes sense. Decrease FAD2, doesn't make sense. And if you look at the direction of the precursor and product, you guess FAD2 should also increase. Because you increase your enzyme, you decrease the precursor, you increase your product. That's the expectation. But in reality, the farming allele is associated with decreased FAD2. And that's something that breaks all the whole picture here. That's why I did not put it in this picture. Is that when you do the same analysis, this allele is associated with higher FAD1 and FAD2. So accidentally, you guess it. And
Same tissues then?
Same tissue. It's very weird. It doesn't fit the phenotypic association at all. And one of the hypotheses we have right now, I don't want to go in, is FAD1, FAD2, they are head-to-head orientation. So they have some shared regulatory region. So may have some competing regulation. Maybe. We're doing the cellular experiment. So the whole cellular experiment we're working on is trying to figure out why the variant is associated with opposite expression pattern in FAD1 and FAD2. Yeah. So it's actually a very interesting question. I don't know how you get there.
Hi, very cool talk. I just had a question about the haplotype bifurcation plot. And I'm guessing, is the evolutionary signal linkage disequilibrium when you compare the derived and the ancestral allele? And does the null model, because I saw the p-values, does the null model account for demographic changes?
Good question. The short answer is no. I did not really develop a null model for that evolutionary analysis. The way I got the p-value was based on an outlier approach. I just do the calculation across the genome and then just use the empirical distribution to see where they locate. So I did not really apply a null model. Good question.
Awesome. Well, thank you so much, Calvin. That was fantastic.
Thank you.